that feeling of mental fog. It might be a structural failure in your brain's membranes. We usually talk about neurotransmitters, but the real signal is happening in lipids, the fatty structures that build every synapse. Imagine these neuronal rafts like tiny molecular scaffolding holding all your ability to focus and remember. If those scaffolds degrade or become rigid due to bad fats, everything slows down. You cannot maintain high-frequency thought patterns necessary for deep flow state because the underlying cellular engine is struggling with membrane fluidity. This is not just about eating healthy. It is microchemistry. Specific PUFAs determine whether your neurons fire cleanly under pressure or sputter out from oxidative stress. The ability of your neurons to function optimally relies less on simple neurotransmitter floods and more on cellular energy management within the synaptosome itself. Think about how sustained cognitive effort demands immense ATP production at peak capacity. Specific fatty acid profiles dictate mitochondrial efficiency inside these brain cells. When lipid integrity degrades, the cell struggles with peroxidation. This stress directly impairs its ability to fire cleanly when you need it most. We are talking about regulating neural resilience through metabolic precision, not just dietary compliance. How does this metabolic integrity actually translate into your state of being? The connection runs deep, influencing far more than just synaptic firing rate. Specific lipid shifts modulate the stability of the autonomic nervous system itself. We are talking about vagal tone resilience. When cellular membranes achieve optimal saturation and structural balance through precise fatty acid metabolism, the entire nervous system gains an intrinsic buffer against environmental stress or fatigue. This microchemistry dictates your body's baseline capacity to manage sympathetic activation versus parasympathetic calm. Understanding this is how we move from managing symptoms toward regulating foundational neurological stability. But interpreting these complex metabolic shifts requires granular data that goes beyond standard blood panels. We can go far deeper than general lipid counts. The next level of insight demands we look at cellular metabolism, treating your plasma as a metabolic fingerprint rather than just a blood test. We need to quantify saturation levels within fatty acids because that precise ratio directly dictates how efficiently your neurons handle stress. This is not about macronutrients. It is microarchitecture. When the balance shifts, key regulatory pathways stall, specifically those involving peroxisome proliferator activated receptors, or PPARs. These receptors act as master switches for cellular energy usage inside every single neuron. They govern whether your cell maintains clean oxidative function under load. This metabolic signature is where resilience is actually coded in. But how do these specific lipid shifts functionally translate across the entire nervous system? Structural integrity of these neural membranes dictates more than just firing speed. It determines receptor quality itself. Think about this connection. Optimal membrane structure stabilizes neurotransmitter receptors across the entire synaptosomal network. But achieving that stability often creates tension. On one hand, perfect lipid composition promotes peak synaptic function and clarity. Yet, when those same fatty acid profiles are subtly off balance, they can simultaneously trigger a kind of systemic neural noise, degrading long-term coherence even if basic cell energy remains adequate. This dual nature, stability enabling flow versus subtle imbalance introducing cognitive static, requires us to look at something far more specific than just saturation levels. We must quantify the lipid raft dynamics themselves, observing how these molecular scaffolding units interact with signaling molecules under stress across different brain regions. The sheer complexity suggests an adaptive cost that seems disproportionate for simple function. We are looking at the most fine-tuned molecular machinery in biology, controlling every single electrical signal across your brain. Yet why would evolution build this level of intricate lipid control? The answer lies beyond local synaptosome efficiency. It involves a deep regulatory loop connecting cellular membranes directly to whole organismal resilience and immune calibration. These specific fatty acid patterns are not just signaling molecules for thoughts. They serve as metabolic messengers that communicate systemic stress levels between the brain, gut, and peripheral tissues. When this lipid communication fails, you are not experiencing isolated cognitive static. You are facing a fundamental breakdown in internal homeostasis across your entire biological system. What happens when those microscopic messages become corrupted? Ultimately, the correlation between cellular health and global nervous system stability is not passive. It is a direct chemical feedback loop you can influence. If lipidic communication breaks down into noise, that breakdown manifests as cognitive fog or dysregulated stress response in your daily life. The specific takeaway here is this: stop guessing at dietary improvements for vague better brains. True calibration demands looking at the precise fatty acid saturation profile within your cells to measure metabolic resilience directly. This moves beyond simply checking blood sugar. It requires understanding lipidomics as a functional metric of neurological architecture. If you want to quantify these shifts and understand how microchemistry translates into systemic clarity, I put all my research on jmthomasofficial.com for the full archive. Consider this final synthesis. True neuroplasticity is not about adding ingredients. It is about precise molecular architecture maintenance. When lipid profiles are perfectly calibrated, they create that rare state where peak synaptic efficiency coexists with systemic nervous system silence. This balance allows for deep focus without crashing into neural noise or chronic inflammation. Your immediate action does not need to be a massive dietary overhaul. It is the disciplined pursuit of metabolic ratio intelligence. Look beyond generalized health advice and begin viewing your plasma chemistry as a direct measure of foundational cognitive resilience. To view the full research archive on how these microshifts translate into clarity, go to jmthomasofficial.com.